Hi, everybody. Tonight we're going to quickly talk about homeostasis and cell signaling and feedback loops that help systems maintain homeostasis. So, to start with, what do we mean by the term homeostasis? I have included a reference to a recent paper that was published in PubMed about how homeostasis is really the central organizing principle of all physiology and maybe underappreciated. Basically, homeostasis is all of the processes that work together to regulate a biological system and help them maintain stability while adjusting to any kind of external stimuli or changes in the external conditions. So, to maintain homeostasis, there's kind of a set of steps that a cell or a system or a tissue will go through in order to achieve homeostasis and to respond to changes in the external environment. So it's going to kind of reference or walk us through this diagram over here on the right. So step one is that, first of all, we have a stimulus that changes this situation from normal. So here we have normal, and then there's some sort of deviation. So that's going to be a stimulus. This could be light hitting the photoreceptors in our eyes. It could be ethylene gas being picked up by a plant cell. It can be a change in temperature. Whatever it is, there's been a change from what our body considers or what the system considers as being normal. The second thing that happens is there has to be some sort of receptor that detects the change. So it could be a pressure receptor in our skin, or again, a receptor in our eyes, or something in our organs that picks up a change in our bloodstream. But that receptor then has some information that it sends along what we call an afferent pathway towards the control center. In humans and mammal organisms, vertebrates, our control center usually is in the brain or in the spinal cord. So the control center determines the appropriate response. And it's not a thinking process, it's a detection of changes in chemical signals in the environment, and then a response that's coordinated. So then the information goes out from that control center back towards wherever the need is for a change. And then whatever receives that information that's coming back from the control center is called the effector. And the effector is whatever is going to cause a change to bring us back to normal. So again, if we look at this little flowchart over here, we've got a normal set point, and then there's some deviation picked up by a sensor that's sent to the control center. The information that comes from the control center impacts an effector, and that causes a correction and gets us back to normal. So here's an example of something that might happen or that you might have experienced before. So I put my hand down on something hot. I accidentally touch the hot plate on my stove or my curling iron or something. There's a temperature receptor in my skin that sends an electrical impulse from the receptors to my spinal cord and to my brain, where there's an interpretation of this input that's coming, and then a message that's sent back through my spinal cord, back to my muscles in my hand, where I relax my triceps and contract my bicep, and I pull my hand away. And that takes us back to normal. At least have removed ourselves from that stimulus. Okay, so the process in which our bodies, our biological systems respond to stimuli can either be what we call a negative or a positive feedback loop. These names can be confusing because in our normal everyday use of these words, negative means bad and positive means good. But that's not actually what's happening here with a negative or a positive feedback. One is not good, one is not bad. They're both responses that our body can help to get back to set point. So a positive or a negative feedback is going to be something that regulates and helps us get back to set point. And a positive feedback is going to be something that amplifies. So those are two words that I try to get my students to associate with negative versus positive feedback. So let's talk a little bit about negative feedback first. So in negative feedback, we have a set point. So in both of these systems, we have a set point that's a normal. So imagine this balance down here is at normal set point. and something happens, or some sort of stimulus happens that causes this to be unbalanced. And when that happens, then the responses that take place in the system help to bring this back to set point. So we're going from out of balance to back in balance, back to set point. In a positive feedback, we're at set point, and then there's a stimulus that takes us out of that set point, but then the response from the system is to continue to keep us out of set point. So to continue to amplify the response, that the original response from the stimulus. So that can be kind of confusing, but let's put that in context with a couple examples. So first of all, we're going to look at an example of a negative feedback. Again, this is regulatory, so we're going to regulate or get us back into balance. So this is how our body regulates our blood glucose system. And so when we eat a meal and we've gone through digestion, then glucose molecules are moved from our digestive system into our blood, and that can cause a rising blood glucose level. If our blood glucose gets too high, that can have damaging effects on our brain and liver and other places, and kidneys. And so a high blood glucose level in systems that are working correctly stimulates the pancreas. Once the pancreas is stimulated, it secretes a hormone called insulin. So it will start the production of and secretion of insulin. And insulin docks on the surface of muscle cells and, well, any body cell, but the body cells and liver. And that causes blood glucose to move from the bloodstream into the cells. When that happens, that decreases the overall blood glucose level, and we go back to set point. So homeostasis has been restored. If the stimulus is that our blood sugar is too low, then our body responds by releasing stored glucose back into the bloodstream. And the way that it does that is, again, the pancreas is the control center in this situation, or is the reception point. So it is detecting the decline in the blood glucose level, and then cells inside of the pancreas are producing a hormone that's called glucagon. Glucagon is kind of the opposite opposite hormone of insulin, where it is actually going to act on the liver and muscle cells to release glucose back into the bloodstream. So up here, when we have excess glucose, the liver takes up glucose and stores it into these long chains of glucose molecules called glycogen. And so when blood glucose is low, this glucagon that's being released by the pancreas actually acts on those storage components, glycogen, and causes glucose to be broken back down, the glycogen to be broken back down into individual glucose molecules. And then those are released into the bloodstream, bringing the blood levels back up and back to homeostasis. And so as soon as that happens, as soon as the blood sugar rises back to normal, then we will actually shut down the production of glucagon. And the same thing up here, once the blood sugar has dropped, then we shut down the response of insulin being produced. So that's why it's negative, because we're reversing the effect of the stimulus to bring us back to that set point. Okay, in positive feedback, or what we want to think of as amplification, this is an example of how babies are born and the positive feedback cycle that's needed to get a baby out of the uterus. So what happens is when a baby starts to grow big enough that it's putting pressure on the cervix, inside the tissues of the cervix are some stretch receptors. And when those stretch receptors get stretched, they send a nerve impulse to the brain. And the brain recognizes that as a stretch and tells the pituitary to produce oxytocin. Oxytocin is a hormone that is released into the bloodstream and then docks on the cells of the smooth muscles of the uterus. And when that happens, oxytocin causes the smooth muscles that make up your uterus to squeeze. It also can impact your digestive system and other places, but causes those smooth muscles to squeeze. And just like squeezing the tube of a toothpaste, that puts more pressure on the cervix. And then more pressure on the cervix, more impulses to the brain, more production of oxytocin, more contractions. And that's going to keep happening until the stimulus is completely removed and the baby is out. All right. So we're going to zoom in a little bit and look at what's happening during these feedback processes at the cell level, because ultimately everything in life happens at the cellular level. So what's going on here is that the hormones, let's say insulin or oxytocin that we saw in our previous examples, are floating in the bloodstream, moving into the interstitial fluid around the cells, and they dock on membrane proteins that we call receptors. So these proteins have a specific shape and they bind to a specific ligand, which is kind of a general term for any molecule that is going to be received by a receptor. When this happens, it changes the shape of the receptor protein, which then can activate other relay molecules. Sometimes they're proteins, sometimes they're non-protein molecules, but things get activated and other processes take place and activate multiple cellular responses. And so this is a kind of a basic signaling transduction pathway is what we call it. And there's 3 steps to signal transduction: the reception of some sort of environmental messenger, the transduction, which is the increase of responses or amplification of an activation of different relay molecules. And then the third part is response. So whatever the cell is supposed to be doing based on that information. So in our 2 examples in the past, the response would be uterine contractions causing muscle contractions, or the response was the uptake of glucose molecules into the cells. So just to kind of explain some different types of cell signaling and how this can work, there are different ways that the signals can be produced and where they end up being picked up. So this first one is called paracrine signaling. This is when a signal is released from a cell and it's impacting a cell that's kind of in the local environment. Autocrine is like what we saw in the video about bacterial quorum sensing, where a cell can produce a molecule, release it into the environment, and then actually pick it up on one of its own receptors to then stimulate more of that molecule to be produced. And so that's an amplification process, and that can be called autocrine because the cell is producing the signal molecule that is also going to stimulate the cell. Endocrine signaling is the process of a gland or some organ producing a hormone or a molecule that then travels through the bloodstream or a long distance to then interact with a cell in another location. Synaptic signaling is also a long distance type of signaling, but this happens between nerve cells. And nerve cells have 2 different modes of communication. They will pick up chemicals, and then that stimulates an electrical response along the length of the nerve cell. And then at the end, it'll produce more chemicals that float across the space until it hits the receptor of another nerve cell. So synaptic is any kind of signaling between nerve cells. And then the last one, juxtacrine, is when we have actual cell-to-cell contact. So a compound on the cell membrane of one cell actually fits into a receptor on the surface of another cell. And then once that happens, the cells can interact and cause a response. So here's some of the responses. So those are the types of kind of reception that can take place. These are types of cellular responses or system responses that we may get. So the first one is a cell division and proliferation. So proliferation basically means to grow more tissues. So this might be for the purpose of replacing cells. This might be what's happening in a system right after egg and sperm have fertilized. The sperm has fertilized the egg, and now we're dividing. We're signaling the cells there to start dividing and proliferating. Also transcription. So transcription is the process that eventually leads to making proteins. So this is making a copy of DNA onto RNA so that we can make proteins out of it. Amplification, if you remember in the fight or flight video that we watched, when there are some systems that release molecules that then amplify the response in other systems. So lots of things are happening. So one little scare and the release of epinephrine, and now we have lots of reactions that are taking place in the system. And then termination of a response is also something that can happen. And on a cellular level, this may look like apoptosis, where the cell actually goes through cell death to stop the process, the stimulus. Okay, so here's another example, simple example of signal transduction. So again, we've got a hormone or some sort of molecule that's attached to a membrane receptor that's activating a relay protein. In this case, this relay protein is opening a transport channel, and we have ions that are diffusing in through an ion channel through diffusion. Then they become a secondary messenger that then activates another relay protein, and then that leads to a response. So again, this is just kind of a generalized example of a system. Here's a more specific example of what happens when adrenaline or epinephrine is attached to a receptor in the cell membrane. So this is representing the cell membrane. And we've got a receptor that's activating this molecule that then activates this molecule, which brings in, can bring in some calcium. We've got some ATP expenditure that's creating something called cyclic AMP. And then there's a production of this molecule. This molecule becomes activated. Anytime you see a little like energy ball there, little sun, it's because ATP or some sort of energy molecule has activated this. This is going inside the nucleus, and it's acting on transcription factors, which then cause transcription of DNA. So it's basically the activation of a gene to start producing some sort of protein that we need for a response. Okay, and if we have changes in the cell signaling processes, whether it's at the receptor level or one of those signaling molecules inside the cell, this is really where we get into the pathology of all disease. So in a previous module, I had you look up some cellular diseases, some diseases that are caused by dysfunctions in organelles. Those dysfunctions probably have some sort of disruption in a cell signaling process like this. So this is a side-by-side example of regular dopamine signaling and one signaling what it looks like in someone with Parkinson's. So you can see in this normal state, we have dopamine that's being produced by the cell and then released into the synapse, the space between these two neurons, and then that gets picked up by receptors on the postsynaptic nerve cell and causes a series of relay molecules to then lead to transcription. Over here, with someone with Parkinson's disease, there's no release of those vesicles. So somewhere we've had something go wrong, and now instead of arrows that are producing, we can see dopamine's being produced right here, but then it's basically being broken down, not being released, and this cell then isn't activated. And in Parkinson's disease, person with Parkinson's disease, they shake because they're not getting the signal information they need to be able to control their muscles. And so they kind of shake out of control. All right, so just one last quick review. These are a bunch of different examples of homeostatic regulatory systems. So our pituitary and our thyroid, we've got neuroendocrine and immune system responses. Our liver has responses. We've got this glucose insulin system and just a basic cell signaling. This is an electron transport chain. All of those things really are examples of cell signaling and feedback loops and the way we maintain homeostasis and respond to changes in the environment. And so that's what it looks like on a multicellular organism, where they've got systems, lots of systems and tissue types and different types of cells and lots of hormone communicators. But in single-celled organisms, we're basically going back to what we've learned about transport. So things that are moving in and out by diffusion, endo and exocytosis, active transport. And then this is an example of what we would call that juxtacrine signaling, where this is how yeast mate. They have cellular receptors and they produce compounds that are kind of recognized by the different type of cell. You can see this purple A and this pink alpha. they're producing different molecules and receiving the molecules from the other type, and then that allows them to fuse together. They actually combine their nuclei to produce a fully adult yeast cell. So lots of interesting stuff going on in homeostasis. So I hope you enjoyed that, and that if you have any questions, let me know, and we'll see you soon.